In this video, we're going to be talking about the initiation of transcription. RNA polymerase is the enzyme that's responsible for copying a DNA sequence into an RNA sequence during the process of transcription. Prokaryotic polymerase has the ability to bind to a DNA template without the help of other proteins. However, eukaryotes are not like that. Eukaryotes require special proteins, which we call transcription factors, to go in and bind to the promoter region and bring in the correct RNA polymerase for the job. Eukaryotes have three RNA polymerases, RNA polymerase 1, 2, and 3, which all have different jobs in the process of transcription. Each RNA polymerase requires a different set of transcription factors to bring the RNA polymerase to the DNA template. RNA polymerase 1 is located in the nucleolus and is responsible for making rRNA, or ribosomal RNA. RNA polymerase 2 is located in the nucleus and is responsible for making mRNA, microRNA, and snRNA. It is also responsible for transcribing the majority of eukaryotic genes. Lastly, there is RNA polymerase 3, which is also found in the nucleus. This polymerase is responsible for transcribing 5S pre-rRNA, transfer pre-RNAs, and small nuclear pre-RNAs. The tRNAs will be used later on in translation, and the small nuclear pre-RNAs will be used later on during splicing. Since RNA polymerase 2 is responsible for transcribing most eukaryotic genes, let's begin there. There must be a promoter region on the DNA sequence that initiates RNA polymerase 2 to transcribe at the promoter. The promoter region is a specific sequence of nucleotides that can bind very well to the polymerase. RNA polymerase 2 transcribes genes that have AT-rich promoter sequences. This AT-rich promoter is known as the TATA box, which is located at -25 to -35 bases relative to the initiation site. Transcription factors are used to recruit RNA polymerase 2 to the protein coding gene. Transcription factors also help influence which genes are used in which cell. Some eukaryotic promoters have a cat box located at -80 bases relative to the initiation site. Getting RNA polymerase 1 and 3 to the DNA template is pretty similar to RNA polymerase 2. Unlike RNA polymerase 2, which favors AT-rich promoter sequences, RNA polymerase 1 favors two GC-rich promoter sequences located in the -45 to +20 region of the DNA sequence. However, genes which are transcribed by RNA polymerase 3 have upstream promoters that occur within the genes themselves. Altogether, transcription requires multiple proteins to interact with the DNA strand and with other proteins. This process of initiation is the first step to completing the process of transcription.